tonight and tomorrow night, we're hoping to present what I was calling it biblical science on a let me try that again. Maybe I should pray first. Father, please anoint and bless and visit with us tonight. And encourage your body, encourage your saints with these thoughts and these facts as we consider the sciences and we consider the works of God. In Jesus' name we ask, amen. Okay, the tongue is working perfectly well now. As I was saying, I was calling this biblical science on a micro and macro level. And so we've got a total of 6 different presenters. Over the course of the 2 nights, we're going to be 3 more or less micro-level presentations tonight. First is Doug Jansen, and he will be speaking on electrophysiology of plants and molecular machinery. Second will be Pastor Mike Klicka speaking on irreducible complexity. And the third will be Dr. Tom Chung speaking on cancer, cells that have lost their identity. And I will ask each of the presenters to give some amount of credentials as God sees fit. But what we're trying to do here at this point, we're not looking to bash evolution at every turn. Charles Darwin's been dead for a good long time, and we are really moving on to the concept of what is God's perspective on the sciences. Is there not some link between the Creator and His creation? Tomorrow night we'll have myself speaking on some of the philosophical efforts that have been made to separate science from Christian faith. Pastor Philippe Siraji will be speaking on a philosophical and logical argument on why the young earth 6-day creation model is the most logical and reasonable and viable model for the beginnings of the universe. And then John Sabo will be speaking about the age of the solar system. So tomorrow we'll be sort of moving into the macro level things, out of the cells and into the universe. All sorts of science fiction mottos from, television shows and movies come to mind, into the universe, but we'll leave it at that. So we'll have 3 presentations tonight, and then we'll take a break somewhere in the middle of it, probably after the second presentation. And then at the end, we'll have until 9 p.m., however that works out, for questions, answers, and discussion. One of the things, if any of you are scientists or have any significant training in the sciences, you know that none of us know it all. There are 2 quotes from Shakespeare that always come to mind when I'm considering the sciences. One is from Macbeth. There are more things in heaven and earth, Horatio, than are dreamt of in your philosophy. So none of us individually has all the answers. So bring what you have to the table, and we'll have some good discussions. And maybe I'll get around to telling you what my second favorite quote from Shakespeare is. But that being said, Doug Jansen.
Sebastian, is this doing its thing? Okay. I have never used this to do that, so we'll see what happens. All right. I know Pastor Phil just prayed. Let's pray. And by the way, I really love what you said. Just built me up to no end. He talked about love and salvation. And I went home and thought about it a lot. I just wanted to say that to you publicly. Okay. Lord, we just commit this time to you. So thankful for your love and your care, the salvation of our souls. If you can save us forever, surely you can give us wisdom for our daily life, wisdom for talking to people about our faith, wisdom for our families. And let's pray for a special covering for us this summer as we just go forward in the vision that you have given to share with our neighbors, to share with our neighborhoods the love of God, and open our mouths to speak about your creation, about man's depravity, about the solution, about our eternal destiny, our eternal home, and give us a love motivation in all of it. In Jesus' name, amen. Amen. This title here came from Pastor Phil. Somehow we were trying to buy a house and I was all stressed out about it. Can you hear me? Is this all right? And he just texted me something like, God does all things well. And that's encouraging, isn't it? I just sat and thought about it. Yeah, he does all things well. And I don't really have to worry about like the details. I mean, I know we do in practicality, but I say God's already dealt with so many things, you know. Like making a planet is a big deal. Like me getting into a house really isn't that big of a deal. And today I signed on the house with my wife and just walked away with a little key dangling, looking at it. And I was suddenly struck with fear, you know. Like I own that thing over there. Roof caves in. No. God does all things well. Really, this, if you were to just think about throughout the whole presentation, this is just spotlighting a couple things. There is an outline to what we're going to be talking about. This comes from Mark 7, and just people looking at Christ and saying he does all things well. And we can let that be our guiding phrase. I could just pose a question to the audience. How close did Judas Iscariot get to Christ in the end, during the betrayal and all that? Do you remember? Like right here. And when the kiss is exchanged, you know, like you go to France, and Americans are always really awkward in France because everyone's smooching each other, and you never know who to kiss and all that. And you know you know what I'm talking about. And the kiss is a very intimate, friendly way of greeting somebody. And it's ironic when we look at Scripture, Judas kissing the face of the one who made him. And just think about evil getting that close to Christ. Spittle, you know, little saliva. And the mouth that was made by the order of God in the history of all the people that led up to Judas Iscariot, all his people that came before him, and then those lips were on the face of the Creator. And I just always think about that scientists maybe are in the discipline that comes the closest to the Creator, looking at the details of his handiwork, and they don't get it. Many don't get it. And that's not an indictment against scientists. I think it's more an evidence of our depravity. We're just lost. You know, Adam walked with the Lord, and then, you know, something goes wrong, and he's like thinking in a crazy way, like these leaves are going to cover me, and you know, let me hide and all that. And we're so silly if you think about it. You know, we're like hiding under the couch with our feet sticking out, and it's very obvious. I remember, you might even know this sci-fi story, but a guy putting a towel over his head, and the animal can't see him because he has a towel over his head. It's a silly thing to think about. And we're like that. We hide from our Creator. Even then, do you believe that God is speaking through science? Is he speaking to scientists? Are scientists going home after a long day's work? By the way, scientists really do work 7 days a week. Many of them, professor I work with at UMBC said, you know, I don't believe in weekends. And I was like, well, I do. I'm mowing the grass, and they really work very hard. Imagine working your whole career and you're just kissing the face of the Creator and you don't get it. And so we really, out of a heart of love, we want to bless and minister to and draw people in understanding science. You're going to see a number of images that are from scanning electron microscopes and things like that during this. That's a seashell. It's not very complex in one sense, but it's an arrangement of calcium carbonate and some other cool things. If you're not familiar with so much with scale, we're going to explain a little bit about that to you tonight. And Pastor Phil, I probably should ask you how long I have because I have no clue. Okay, all right. I'll try to be quick. These are 4 things that I like to work on and I'm interested in and I'm studying. If you really like to geek out on your own, go online, scholar.google.com, and just read scientific papers. And you say, well, I'm a construction worker, I'm swinging a hammer all day, and I say, go home and read a paper, and you'll read recent research. It's available to you. Don't be intimidated by it if you feel like doing that. Sometimes they also distill it into easy-to-read journal articles or summaries. One thing that I'm working on right now is plant electrophysiology, which I know that that's part of our talk, but I won't talk very much about it tonight, except to say that plants have something like a nervous system, and they can feel, they can sense things. I'll give you an example. At the edge of a forest, if a fire is approaching, on one side of the plant, there's actually an electrical impulse that travels up and down the xylem and phloem. There's an electrical current carried, and this triggers the release of chemical signals that are blown through the rest of the forest. And we actually see the closing of these cells called guard cells all across the forest, all initiated by an electrical response from just a few trees at the edge of the forest. So interesting things programmed into trees. Trees don't have a central nervous system. They really don't have any brain. We know that. Trees have never asked me questions or anything. Why are you chopping me down? But they feel, apparently, they have some sort of sense of feeling, and they understand when they lose water pressure and that kind of a thing. But there is actually an electrical current present in plants, and the Department of Defense is very interested in that. That's how I'm funded through a grant to research if we can put these plants in airports, and if they sniff something bad in the airport, and no one would know that it's just a plant there, but if the plant sniffs something bad, it closes a leaf or something. Mimosa pudica is a good plant to look into. When it gets irritated, it closes up. And if we could program plants to do that, then subtly, without any great alarm, we could sense things like chemical agents in an airport or train station. So that's kind of a cool thought. Plants aren't that dumb. I look at my house plants very different now. I have like no green thumb, and you can ask my wife about it. People give us house plants, and I'm like, oh, I'm so sorry because it's going to die. But they seem very resilient, even my best efforts to kill them. The other is insect biophotonics. Another DoD project right now is to put chips into the backs of beetles and a little CCD camera on their chest and fly them into battle zones. They're extremely small. They can be remote controlled right now. So you can make a beetle go left and right and tip and tilt and all of that. And they power their own circuits because they're eating things and all that. So you can dip them in some nutrient solution and send them out into Afghanistan. And they just look like a bug flying around. But yeah, so insect biophotonics is a really cool thing because they also can mimic human biophotonics, how light can interact with that stuff. They can be controlled with light pulses. Even right now, mice traveling around, they can put a light probe in the brain and turn them left and right. Scary because they would like to go to the human model at some point for therapy. Just throwing that out there. Light neuron interactions is really hits close to home. My grandmother has Parkinson's, and she has had 2 deep brain surgeries, which mean that they basically take the top of your skull off and they implant a probe. You might even know about this. You might have a relative that has had this happen. And there's a probe that goes down deep. Her last surgery went very bad, and she lost all her memory and all her speech centers for about 4 weeks, and then she finally started to remember who she was. We would like to move away from the invasive model of cutting people up and instead be able to send in light into a specific nerve center and to initiate a response, get the brain to be rewired or whatever. And that has some very promising future thing. I'm sure down the road it will be a part of normal therapy. Right now it's kind of on the fringe. And the last, I kind of mentioned this, detecting IUDs. I work in a research group at UMBC. This will be the 7th summer. I'm funded by the National Science Foundation through Princeton University, and we almost always work on quantum cascade lasers, and that's to do standoff detection for improvised explosive devices. I wrote IUD, which is not correct. It's IED. Sorry. Yes, there's quite a difference. This is a poster, and of course, I know a lot of science students do this, and it's very useful. I'm not going to explain everything on here. I can't even read it from here. But long story short, I spent a summer doing brain surgery on ants, and I have high school students here, so I've said that to them before. But brain surgery on ants is very difficult, and my professor gave me a Dremel. If you know what a Dremel is, it's not used for brain surgery ever. And he said, just go with this. So I spent about 7 weeks trying to figure out how a Dremel could drill into the brain of an ant. And if you want to know more about that, ask me later. But on the right side there, something that just looks like a bunch of random graphs, but what we see there is actually light. Over on the left, there's something pink. Can you see that? And there's light scanning across a neuron. Okay, so on the right side, it's horrible, but there were bugs super glued down to a platform, okay? And they're looking up at me. When they come out of anesthesia, they're like, why am I stuck here, and what are you doing? And we scan their abdomens to see if we can trigger nerve responses, and we have probes in their brain that can see if there's a nerve response based on different kinds of laser light. And long story short, there's a lot of bugs that are sensitive to laser light scanning their abdomens. And we discovered there's some bugs that are specifically sensitive to infrared, and they actually seek out forest fires. They'll go to a plant that's on fire, and they'll lay their eggs there temporarily and leave really quick because their eggs can only hatch at 400 degrees or something crazy like that. But I said, hey, maybe there's some bugs that are not fire-loving bugs, but they would also be tickled by light. And so you'd be surprised how we got these bugs. It was me outside with a net, It was very embarrassing outside. There's people inside doing real research, and I'm running around the parking lot, catching things. That's great. No, I did buy some also, but anyway, you can talk to me about that later. Long story short, our nerves are very sensitive to light, and specifically coherent laser light. So it'd be a great thing if we can stimulate our nerves with light. This last summer, I spent time trying to develop remote sensing networks with trees to set up some sort of a response where we can tap into the natural response of a tree when it's sensing, as the picture shows there, a fire or something chemical, but to be able to put a little detector inside of a tree. The Weather Service loves to know what's going on everywhere, and of course other arms of the government do too, but the Weather Service would like to know, is it going to be a forest fire in this area, or dry conditions that could lead to a forest fire? But putting a sensor out there is very, very expensive. Replacing batteries, very, very expensive. And so if we could power it all by tree power, that would be nice. And it turns out we hooked up tree limbs like batteries, and we're able to extract enough energy to start powering devices, which would be really neat to be able to plug into a tree. I'm not saying we're going to power your cell phone anytime soon, but there are some businesses actually. You can look up Voltree is a company that is starting to sell this idea. Anyway, that's over on the right side where there is some purple bars. This is actually laser stimulating response from plants. We scan across a plant tissue and are able to see an electrical spike. So that's kind of cool stuff. I'm sure some of you recognize what this is. All right, this is what I'm actually going to talk about. I want to talk about how small is small. Okay, a lot of people know orders of magnitude and stuff like that. I'd like to try to kind of reintroduce you to that. Look at that, my email thing is popping up. My wife will send me some embarrassing email. Famous molecular machines. These are something I'm just going to spotlight and not maybe talk in detail about, but give you some, maybe we stir up some appreciation for design elements. ATP synthase, we'll talk about. Bacteria flagellum, not very much, but some. And that's the picture there of something flapping in the breeze. Bacteriorhodopsin, which is an interesting molecular machine. Chloroplasts, which harness sunlight. And the nuclear pore complex, as well as some random things that I thought were cool, and finishing off, okay? So I think we can follow that together. Do we love the Bible in our church? Yeah. We love the Bible. I tell my students I got a great biology book here, and it's all going to burn. My chemistry book's all going to burn. Everything nice is all going to burn. Fervent heat, you can look it up, 2 Peter. It's going to burn with fervent heat, even the elements, you know. All the chemists are bummed about that. But the Bible is going to remain. It's going to remain. It's going to persist into eternity. And I love this Bible verse because I am one of those foolish things, I believe. I am. I think so. If you ask my wife, she would confirm that. One of the foolish things of the world. God has chosen foolish things in the world. If I were to ask you to simply look at the pulpit here, which is made out of wood, and estimate how many cells were needed to make the top of this pulpit, I could prove to you that you are confounded, because I don't know how many of you can estimate that. Also, how many of you can make wood, like make it? Can you make it? Pastor Phil loves to process wood, but make it, okay? It's a little tiny thing, and it confounds you, okay? I have 3 kids, they're also small, and they confound me daily. The weak things of the world confound us. And I love the point of this. It's not to depress us or anything, but it's this, that no flesh would glory in his presence. I actually kind of think like Judas with Christ might have been like, you know, just the spirit of the devil there is just like, I'm winning, you know. But actually, did the devil win? He didn't win. But where was Peter? He's like, you know, warming himself by some fire, foolish, confused, small, weak, and we just had Pentecost. Think about what Peter was doing, you know, thousands of years ago, going on with the Lord, right? With the Bible. He was a foolish, weak thing. Many foolish, weak things in the room here. Bacteria on the ends of your fingers can make you very sick. Ever heard of E. coli? Okay. Little kids playing around in the muck of their yard, getting sick. Little foolish things can really affect us. HIV. Are you scared of HIV? I think a lot of people are. Ebola virus, right? Just a breakout a few months ago in Africa. Little tiny, tiny things can confound us. Okay. Recently, this was in the news, a mite, tiny little mite, was found to be like proportionally the world's fastest thing. And this is a little chart here. It just says if all these animals, human, cheetah, sorry, humans aren't animals, but a human, cheetah, horse, a black iguana, and a mite, if they were all compared at the size of Usain Bolt, by the time Usain Bolt runs 100 meters, the mite would have run 6,130 meters, or more than 6 kilometers. Okay, it's a little mighty mite. Is that confounding us? Yes. I mean, The researchers that were looking into this, I read their paper, and I think they were in shock. Like, this little guy can run that many body lengths, even though in real life he only runs half a mile per hour, and you could easily outrun him. If he were your size, he would be winning the Olympics. Small things confound us. How small is small? I'd like you to consider a water droplet. A water droplet is very small. The scale is given here. We are kind of talking tonight in my talk on the nanometer scale, so really, really small, but not the smallest thing. Down on the femtometer scale, there are some things that really, I mean, humans are jumping into this field of smashing particles together and figuring out what quarks are and elementary particles. If you don't know what a quark is, if you're still wondering how you walked into the room tonight, thank you for coming, by the way. Quarks are part of a family of subatomic particles here, elementary particles. You might recognize things from high school like electrons. Does that ring a bell? Electrons. They are there on the little family of super awesome things that make up the universe. If you want to know more about that, you can look up things like the Large Hadron Collider and other accelerators around the world. Anyway, we're not going to talk so much about that. We're kind of up there on the nanometer scale, somewhere water molecule sized, a little bit above that. But just to know that small gets really small, all the way down to something called the Planck limit. Just a quick note about quarks. This is from Fermilab, which is, I guess, America's best accelerator. And a few funny things that they say here in this magazine article. Okay, quarks are called the fundamental building blocks of matter, and they're like God's Legos, maybe, something like that. I love how there's such a resistance in science to say anything supernatural. It just says nature designed 2 copies each. This is a recent article, relatively recent article. And there's a resistance to say anything supernatural, but even in an accelerator magazine like Symmetry, the word design just comes up. And I'm sure somebody took it out later, but this is a screen capture from their website. And just to think about the design at the smallest level, sub-femtometer scale, there is a symmetry. This is hence the name of this magazine this came from, Symmetry. There's symmetry at the sub-femtometer scale. Okay, a few points about molecular machines. If you were in my high school chemistry class, I'd ask you to write all these down, but you're not. And this is not about M&Ms, but this is about molecular machines. These are nanoscale assemblies, really, really small things that do stuff. Okay, that's how I explain it to my own kids, that there are little things in nature and they do stuff. And my daughter says, like what? And I would say, all right, try to explain it to her. Well, honey, there's the sun, and then by then she's already walked away. But anyway, molecular machines, for instance, can harness sunlight and do cool things with it. They exhibit a high degree of ordered arrangement, unlike my kids' rooms and my own desk. Molecular machines are biologically important. They are necessary. Right now, even in your most, after work, tired state, your body is generating a lot of energy, batteries called adenosine triphosphate, and that is all the product of just one of many molecular machines. Molecular machines can work together and produce big effects, and that's awesome. I don't know if there's sound here, and even if there's not, it's okay. I wanted to just show you one of these, and we'll see if it plays.
Complex energy they create are key aspects of the biological world. A good example of the use of a gradient occurs in the mitochondria when ATP is synthesized. ATP is synthesized by ATP synthase, a large complex of membrane-bound protein. Here we see ATP synthase, along with other membrane-bound proteins. Notice the large difference in the number of hydrogen ions on the 2 sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP.
We'll just pause there. Just showing you a little clip here. This is from NDSU's Virtual Cell. It's a great website to go see cool stuff like that. Do you notice it's a ratcheting mechanism? It's unidirectional. It's concentration driven. If you were looking at this on the macro scale, if I had it in front of me, you'd say that's just a man-made device. It's a gumball machine. It's a whatever, a lathe or something. You know, humans love to build machines. But this is extremely small. On the nanoscale, extremely, extremely small. And these are littering the wall of the mitochondrion. Mitochondrion is like the power factory of the cell. A little bit about ATP synthase. This is actually a quote from a Answers in Genesis, which you guys may be familiar with. And they simply say that setting up this machine would require nature really putting things on pause at thousands of different points in what evolutionists call natural selection, and keeping the best things in a high energy state in order to get this thing to work. I don't know if you've ever tried to harness 20 kindergartners, okay? Anybody ever done that? Nora is an expert at, she's got all the little kids holding a little string and they're walking down the sidewalk. Keeping them just in line or doing one thing, or just coloring with one crayon is hard. Keeping things in a high energy state over the course of thousands of years to get it to stick is very, very unlikely. And so it's commonly understood now that ATP synthase probably happened in a very, very quick amount of time. Personally, I believe that it happened extremely rapidly, for instance, in a moment. At the end here, it says new machines are not made by simply adding energy to existing machines. Okay, adding energy to your bedroom could be like rolling a grenade in there. That adds lots of energy, but it doesn't really do a lot of cleaning up. Not that I've ever tried that, but there was an incident in high school with an M80 in a toilet, I'm just saying. So energy does not necessarily organize things, okay? Energy is useful, but it is intelligence that is needed. Actually, if you walk away tonight with very little about all the detail, it'd be great to have in your mind that there is an intelligence. And when I talk to my neighbor about God and he brings up science, I want to mention to him, hey, what do you think about the intelligence in nature? Let me talk to you about some of these things I don't even know about. You ever heard of ATP synthase? The guy's like, oh yeah, I know about that. And you say, well, I don't really know about it, but I know it's intelligent. And where does intelligence come from? Who wrote the books that you like to read? Well, an author. Okay, who wrote creation? And it's hard to continue that conversation without it getting spiritual. What did Pastor Schaller say? Something like getting deep with somebody, pumping gas, and say, what fuels you? Okay. This is when my wife in the car does this when ATP synthase is irreducibly complex. We're going to hear about that later. It's one of these things that had to happen all at once. This is just a quick animation. I'm not going to actually show very much of this, but this is a 3D modeling of ATP synthase working. At the bottom there you can see it's a membrane inside the mitochondria. That's the ratcheting component. It's a catchy tune because I've watched this a number of times. It was in my head later. Not gonna lie. Long story short, these are moving parts. They are all biologically important moving parts. These are assembled ribosomally, and they are collected together enzymatically, and they are doing stuff. Okay, when we see moving parts doing stuff, we start to think that this is a planned design, right? If you found something in the forest that looked different than all the plants around it, if you found a cell phone in the forest, it's very rare that a person would say, man, this forest grows cell phones. It's awesome. I got to come here more often. You sure? That's not how I got my cell phone, walking around the forest. True story, Finland, 2 years after I was there in the forest, came back to the same forest and found the same hat my daughter was wearing sitting on the same rock she left it. Just tells you how lovely Finnish forests are. There's no one bothering anything. Okay, sorry. This is the product of that machine. This is a biologically important molecule, probably one of the most important ones in the human body and in all of nature, and that's adenosine triphosphate. It is an energy currency, as it says here. This is actually a selection from the Nobel lecture from 1997, and Dr. Paul Boyner was awarded that Nobel Prize researching ATP synthase and what it does. Long story short, it's a really awesome molecular machine, and he was hoping that more people would research it because that little tiny thing was super complex. And we're glad for his work, and many people are still working on just that one little molecular machine. I like this thing at the end here. It says, every day an adult converts a quantity of ATP corresponding to at least half his or her body weight and nearly a ton during a day of hard work. That means, okay, first of all, like, I don't make a ton of anything in my day, let alone I don't know. But my body recycles ATP. It's broken apart, put back together, broken apart, put back together, a ton in a day of hard work. Okay, how many of you did hard work this week already? It's only Monday, and some of you were like, whew, 2 tons today. Your ATP synthase engines are working. Wipe out that little molecular machine and you'd be a wreck, okay? We're thankful for molecular machines. This is a pretty SEM. I just looked at it for a while. My daughter said that's gross. It is, a little shrivelly little guy here. I'm very thankful for Dartmouth's scanning electron microscopy lab where I got these images. This is what causes cholera. Anybody want to have cholera? No. Okay. This is one of those little guys that confounds us, okay? And at its back end, if you can call it that, is a bacterial flagellum. And it is a multi-component rotary motor, very similar to what we have in all sorts of motors that are man-made, except way more efficient, about 2 orders of magnitude more efficient than humans can make their motors, okay? When you feel a motor and it's hot, that's all wasted energy. You feel the hood of your car and you're like, yeah, I've been racing around. Well, that's kind of shameful. You feel the hood of your car, that's all wasted energy that you're feeling as a hot car. These folks have incredibly efficient tails, if we can call them that. And they are a subject of a lot of research. This is what happens when you leave a lot of them around with resources and they make more of themselves. And this is a quote from a paper, actually a relatively recent paper, where they call it an extraordinary nanomachine. And honestly, I thought about that. I know it doesn't seem so significant, but I thought about the guy who goes home at night and he says, I researched an extraordinary nanomachine today. And maybe he goes to bed, maybe he's visiting, by the way, I think 77% of after bachelor's programs, 70% are from other countries. And out of that 70%, I think 90% are from China. So Chinese people are coming to America. They're researching things like this, and I've met many of them. And you'd be surprised that many of them that I've talked to have never thought that there could be a designer behind the design. campuses are a great place to talk to people about this. To say, are you researching an extraordinary nanomachine? What a great sign to hold up on a campus, you know? And have somebody what do you mean by that? And say, do you want to talk about the designer of extraordinary nanomachines? Just something to think about. This is a selection of the paper by the same people here. And they have used cryotomography, basically cryo-electron tomography. And that's to take something to a really, really cold temperature and put it on pause, okay? And when they put this flagellum on pause, when it's super, super cold, they can start to image it. And I don't know about you, but if you look at the pictures at the top there, if I had to draw that in art class, you know, it just wouldn't come out that precise. This is a set of proteins that make up the motor that is the bacteria flagellum, and it's high precision. I don't know about you. Does that look high precision to you? Yeah. It's high precision. It's self-assembled, and also it's self-attenuated. It knows when to stop building. So it's great that humans also know when to stop growing. There's some genetic mutations that can cause the body to just keep growing, and that's a bad thing. Our bodies are programmed to know when to stop growing, and much of biology is programmed to know when to truncate at some level of biological advantage. This is an awesome little device that can make the bacteria be able to be motile and move around. Some things actually have multiple tails, and this is another cryo-electron tomograph of multiple bacteria flagellum. Imagine what it would take to get these to not tangle up, okay? To get them to be afraid of each other's surfaces. In other words, they wouldn't get tangled up. To have them running in sync, to be able to feed them enough ATP to keep moving over the life of this small little creature. I like looking at the design over on the right side. Just very symmetrical, very interesting. I don't know. What do I know about that? I'm not these guys. I haven't researched this, but I just look at that and say, 50 nanometers. Are you kidding me? That's just super, super small. If you're not so familiar with scale, our hairs are about 100 microns-ish across, and micron is way bigger than a nanometer. We'll just leave it at that. So this is well under the size of a human hair. I mean, just a small fraction of it. Okay, next one, Bacteriorhodopsin. This is your homework. Go home and try to say that like over and over again. Bacteriorhodopsin. Our eyes have a kind of rhodopsin that respond to light. Rhodopsin family of chemicals respond to light. Bacteriorhodopsin is, as it says here, a compact molecular machine that pumps protons across the membrane. And long story short, it responds to green light a lot. The assumption in evolutionary biology is that all these little bacteria swimming around in a little warm pond somewhere loved to get green light from the sun, and they responded to that. And somewhere along the way, plants forgot to respond to green light, and they started responding more to purple and red. And it's poorly understood the flow of how that would happen, but Bacteriorhodopsin is still around. It's a molecule that's still around, and basically it's a purple pigment, and it's found in plants or plant-like things, not plants, that responds to the center part of the spectrum from the sun. I'm not going to talk so much about it except to say that it is a light-driven process. When light hits Bacteriorhodopsin, it moves. I think that's cool. This is a picture of that, or a little video of that I can show.
Bacteriorhodopsin is an abundant light-driven proton pump found in the membrane of Halobacter halobium, a purple archaeon that lives in salt marshes in the San Francisco Bay Area. Bacteriorhodopsin is a multi-pass membrane protein that traverses the plasma membrane of the cell with 7 long alpha helices. The helices surround a chromophore, retinal, that is covalently attached to the polypeptide chain and gives the protein and cells their characteristic purple color. Retinal is a long, unsaturated hydrocarbon chain that is covalently attached to a lysine side chain of the protein. When retinal absorbs a photon of light, one of its double bonds isomerizes from a trans to a cis configuration, thus changing the shape of the molecule.
Okay,
The
we'll just release you from that. Light makes something happen. Isn't that great? Isn't light awesome? Ask a scientist sometime what light is. Well, let me tell you. It comes in different colors. And no. What is light? Yeah, why does it bend when it goes around massive objects? I thought it doesn't have mass. Okay, just ask your local scientist.
What is light?
I said ask your local scientist. Security. Chloroplasts are, actually there's pictured here, this is from an article about Elodea, and chloroplasts are really awesome little things in plant leaves. By the thousands and millions in plant cells, it's the site where light from the sun is harvested and made useful on planet Earth. Okay, you still with me? Everybody all right? Light from the sun, security. Light comes in, and plants are harvesting this, and also phytoplankton in the ocean. And chloroplasts are very, very biologically important. Honestly, we could argue that you wouldn't exist without chloroplasts, and you say, well, I don't have any green skin or anything. But what you eat needs these, okay? You need to get energy from somewhere. Really, the sun is the source of energy here on Earth. We need something to harvest that, because you put me outside all day long and I'm not going to get enough energy from the sun. I might bake and get a terrible sunburn because I don't tan, but I'm not going to get energy from the sun that's useful. But plants do all day long. They're just sitting in the yard, they can't move around, and they're just soaking up sunlight. Blue and red portions, not green, like we just heard from Bacteriorhodopsin. Amazingly efficient. It does certain reactions when the light's available, and then switches to doing dark reactions when the light's not available, and that's amazing. Picture of some plant cells here. Doesn't it look like bricks? It's efficiently packed in there, and it's amazing. This pulpit here is made of many of those that have died. On the bottom is a TEM image of sets of these, and this is incredibly small. Again, on the nanoscale, and we're looking at stacks of discs here from chloroplasts. Just throw this out there, a little chart. You can see chlorophyll comes in a couple different varieties, and chlorophyll loves to absorb blues and reds. Carotene, like you see in carrots and stuff, rejects orange, actually. Yeah, we do see chloroplasts are incredibly efficient. I'm sorry, I'm never going to talk about that, but this is a light-driven set of reactions. And if you want to know more about that, you can ask me later. There's a Z scheme here, or just a set of light-driven reactions that chloroplasts participate in. You'll notice actually that the scheme goes from low energy up to an excited state and then back down. Lots and lots of things do that in nature. And I love seeing what happens when we excite things and then we let them relax. That's the whole science of spectroscopy is about that. This is the last of the molecular machines. Nuclear pore complex. I don't know if any of you have seen some of these intelligent design films, but they have often highlighted little doorways in the nucleus opening up and genetic code coming out, and they look kind of cool. And I sat there one day thinking, man, what is that? What is the actual mechanism of that? And began to read papers about it. This is from a quite well-known research group, and they have done the same thing with cryo-electron tomography and looked at what does the MPC look like in real life. And they have found that it is a complex set of molecules that work in tandem. You're looking at a scale bar here at the bottom. The doorway in a nuclear pore is about 60 nanometers. Incredibly small little hole through which information is shuttled from the inside of the nucleus out to make things. And if you want to know more about RNA and how that works, I think that may be elucidated some other time in these talks. But long story short, information gets shuttled out through a little doorway that is definitely programmed by information. Selectively allows materials in and out of the nucleus. Okay, so not everything can just like fall out of the nucleus. There's an intricate tagging system to identify things going in and out, and also it facilitates transport. There are some things, you should know about this. There are some things out there where people say the nucleus of cells, really used to be the old cells, but then they sort of got swallowed up by bigger cells. And same with mitochondria, they sort of got swallowed up by big cells, and now they're all sort of like subservient to a big blob. And nucleuses used to be the old cells, okay, and it was those that were in the little pond somewhere. Amazingly, though, the membrane-bound proteins are very, very different, and we're just discovering what they're like. Highly advanced for this purpose. This is not like a dumb, proto-cell. This is a highly advanced mechanism in the doorway of a nucleus, and I have trouble believing that this was somehow some, precursor cell. This is what it actually looks like. This is just a selection of a nucleus, just sort of like a cross-section showing where they are, and they're just plugged in all over the nucleus. Again, this is another really cool picture of a structure. This is a door that opens and closes based on a tagging system. Not everybody can get into the White House. You got to have the right credentials, right? And getting in and out of the nucleus also is a credentialed process. Isn't that neat? Is this a small little thing? And sitting there on the screen is something that's confounding me and maybe confounding you. Okay, this is where I get to just talk about things that I think are cool. What is that? Anybody know? You might know. Don't be humble. It's kind of gross, isn't it? It looks a little bit gross.
Looks like a sac of protein, like a Golgi apparatus.
This is bigger than the cell. This is on a scale that's bigger than the cell. That post in the middle there is about 1/5 the width of a human hair. And it's actually jutting out at you. You can't see that, but it's jutting out at you. Let's look at a few of these structures and see what they really are. These are multicellular things. Okay, so there's a couple cells here. Those are actually stomata, the doorways to leaves, and they let plants breathe. Each stoma is about 1/5 the width of a human hair. Can you imagine that? And they're just littered across the bottom of leaves. You ever notice leaves are a little bit like more green on the top than on the bottom? That's for multiple reasons, but one of them is that there's tons of doorways on the bottom of the leaf where it's more advantageous to breathe. Another item we see here is this little guy, and he's a trichome. Leaves have hair. It's kind of weird. They're not like our hair. They're not made of keratin or anything like that, but plants make these to hinder the wind coming across the leaf. And how a plant ever thought of that without a central nervous system is beyond me. But the trichomes grow on the plant leaf to inhibit the wind ripping water out of the plant, and that's one of its purposes. They protect plants. Isn't that neat? This is a very tiny surface area. What you're seeing here on the screen is about a square human hair width, okay? So like just take one of your hairs and look at it. There's a complex world on that scale. This is a gross part. Okay, I was looking at this and I got really grossed out because in the doorway of a little mouth on the bottom of a leaf are some bacteria like living there. And I just got grossed out. I thought, if there can be that many bacteria in a little mouth on the bottom of a leaf, 1/5 the width of a human hair, I started to just want to wash my hands. It was gross. And I got kids, and my daughter has a cast, which means she can't wash her hand, and it's all nasty. And I started to think about, okay, yeah, I just can't think about that. Here's another guessing game one. What's this?
MC
Is fish blending together or something? What is this? Anybody have a guess? It's definitely hexagonal. I'll put you out of your misery. Anybody want to make a guess? In the inner ears. It could get there, possibly. This is fresh snow from New Hampshire. I just thought it was a cool thing. Cryo level scanning electron microscopy. So really, really super cold environment. The researcher, I'm sure, was in a cold place and brought some snow in to image. Normally, you have to evaporate gold at a high temperature onto a surface, but in this case, they use a super low temperature. And I thought, snow, I'll never look at snow the same way. The post there is about 1/100th the width of a human hair. So we're looking at a structure that you and I wouldn't even notice. It's not a snowflake, okay? Flakes are big. We're talking about molecules getting together and building really cool hexagonal cylindrical structures. We miss a lot of that, you know? And it's not like God's in heaven like, I can't believe they missed that super cool thing that I made. But I really do believe that here in this time in history, as humans get maybe more and more wicked and more twisted in their brains, they also want to know a lot more. Remember Adam and Eve were like, we can know more? Really? Super. Here, have the earth. Knowing things is like a really big deal, right? Monkeys are kind of like, you know, okay, you know, they like want to make more monkeys and get food and throw things around the forest. But humans love learning. Look at you sitting at a table after a long day's work, okay? So this is a challenge maybe from God. Look at my little things and be confounded. Talk about it. Look at the order in creation. I wonder what this researcher went home is, look at the snow outside. I'll never look at it the same way. Beautiful hexagonal structures. Scanning electron microscopy was actually pioneered in part at University of Maryland, Baltimore County, and some of the earliest images of the cell were done there. And there's some pictures. This is an animal cell, just a slice of it, and you can actually see the ribbons of the Golgi body. How many of you ever packed a lunch for little kids in the morning? Packed a lunch. Put the peanut butter and jelly in the bag. True? You ever get a peanut butter and jelly in a baggie or something? And packing it up, the Golgi body is doing that kind of thing, packaging up things in baggies. And the cell is a very active place. This is one mitochondrion featured here, which is where ATP synthase lives and does its thing. Super cool that humans can do this, right? Imaging things and looking at stuff. I highly recommend it. Okay, just a brief summary of some of these little spotlights that we were looking at. I guess every person that you meet, first of all, our motivation talking about science is love, not to bruise people and to beat them over the head with things. But I said this to somebody recently. I said, you're really great at asking questions, but you cannot live a life that is driven by questions. Because if you're just a questioner all your life, you're an irresponsible person because you refuse to come to a solution, okay, an answer. Is everything okay? Something's beeping. A question is important only when I'm seeking for an answer. How many of you have met somebody and with every question they follow it up with another question? And it's like, okay, does anything stick to you? Are you a Teflon human being? Or can I really throw something at you that stays with you? And I love those kind of conversations when you're talking to somebody and you just see in their face like they just got hit with the love of God and they're just never going to be the same. We need to ask questions like, hey, is everything around us the result of random chance? Are these little tiny molecular machines the result of random chance? You have to make a decision about it. You can't say, wow, that's interesting. Okay, say that for a certain amount of time, but then come to a conclusion. Do you agree? Like, dating somebody for 10 years, it's like, you gonna marry the girl? Okay, anyway, sorry. It's just, I'm sorry. Anyway, come to a conclusion. Is it the result of random chance, mutations, non-directed process? Could be just as likely to mutate positive as it is to mutate negative, as it is to linger at a median value. Can new information be generated by this random non-mediated process? And how can we stick the good changes when they're just as likely to fall apart? That's something that you can talk to people about. Say, hey, natural selection is awesome, but we also lose a lot of information when things die off. And what if the weakest thing dies off and we lose their genetic line, which actually had a really positive hidden benefit there? I have heard that we are something like maybe 15 mutations off from being able to generate our own vitamin C. Okay, can't do that now, but if you look at the structure, we may have lost that. We used to be able to do it, and I think many other things. I'll just say an opinion thing, and you can just shoot me down all you want, but I believe some savants in the world, people that can look at the city and draw it in like 5 minutes, are a little picture, a little window into what humans may have used to been able to do. Okay, you can, if you don't agree with me, it's okay, but I think a lot of our DNA has been lost as a result of random changes and stuff. By the way, I think 2,500 years of intermarrying before God said no. So there was maybe a lot of loss happening there, and I think God allowed it so that we weren't like superhuman, awfully evil people. Isn't it great to feel like we're like the dumbest generation? I'm sorry, it's true. It's not like we're getting better. I'm sorry. You can tell by this presentation. Or is it intelligence derived? Did God make it? Did he plan it? Is it pre-planned with a purpose and not just to be cool, not just to be awesome? Did God make water freeze and throw its elbows out in a certain way so that not all the fish die as water is freezing from the bottom of the ocean up or something like that? If you understand what I'm saying, water actually is more dense at 4 degrees than it is at 0 degrees, which means that it freezes at the top and doesn't fall down. You ever notice your ice cubes float and stuff? Water freezes at the top. Imagine if it froze and then fell, and froze and fell, and froze and fell. We'd have a lot of dead fish. Is that hexagonal structure we just looked at actually is a result of the freezing of water and its little hydrogen bonds making ice and stuff like that? Is it pre-planned with a purpose that water does that? I believe that nowadays there is a challenge for even the brightest minds or people that think they're so bright, and God is leaving many, many little Hansel and Gretel crumbs for them to pick up and follow to him. Will they kiss the face and miss the whole thing here, or will they, like Peter, maybe shy away, but then come back and say, I thought about it, and I want the person, not just the thing that I'm working on. I'll leave you with this. I talked to a very dear friend of mine. He's a Chinese non-believer researcher, and he said the longer I'm in grad school, and he's a postdoc now, the longer I'm in grad school, the less I know about anything else. He said, I know about one kind of laser growth in one kind of machine in our lab, and if I ever got hired to do something else, I don't know what I would do. I know nothing about politics. I know nothing about religion. I've never had a girlfriend. I know this is a minority maybe amongst researchers, but he said, I feel like I'm being trapped in a little bubble in science. And I don't think that's from God. Personally, I don't think that's from God. Can we be broad scope? Can we speak to all kinds of people about the gospel? Don't you think there are people like him everywhere? I'm in my little bubble, I climbed the corporate ladder, and now I'm constrained into nothingness. Let's talk to people and say, God's got challenges for you, but it's to lead you to solutions, to conclusions. Correct? Just a little thing left here. This is a man-made mirror assembly from Sandia National Labs. We are making things now on the nanoscale, not just saying, oh, look, a cool thing in nature. We are making them. We are making mirror assemblies that can change the color of the paint on the wall or something like that just by changing a mirror. If it weren't so stinking expensive, we could click a button and make the whole room green, and all the interior designers everywhere rejoice. The last thing here, I'll just say, with every discovery, man can glorify the discovery, or he can give glory to who? To God. The Nobel Prize, as much as it's amazing, is very much to the person, right? Millions of dollars, and then what? But we give glory to God. Maybe nobody knows about us, but that's really where it belongs. That no one, no flesh would glory in his presence. Amen? Amen. Okay, thank you.
The clip-on mic has to get transferred over to Pastor Klicka. So we've got a couple minutes, and I'm going to take my opportunity, because I'm the one with the microphone at the moment, to point out one of the things that Doug said fairly early on. He said, getting high-energy particles to persist in an orderly state for long periods of time is very, very unlikely. And by way of personal testimony, with having grown up with classical public school education, secular training in college, and so on, as a scientist, as a biologist, and then getting saved late in my undergraduate studies. God and I sort of had this conversation regarding evolution and such things as, okay, God and I agreed to put that on a back burner for a while. And when he decided to nudge it up to the front burner and turn the heat up on it, what he brought to my mind as a scientist is the importance of probability and statistical significance. If you took the time to read the research papers that Doug was showing some slides about, you would see that almost every scientific study in the hard sciences, and especially in the biological sciences that I deal with, somewhere in the methodology and in the results, you would see mention of the statistical analysis. In other words, each time we observe an event take place, a phenomenon, we have one datum, one point of information. And you and I are used to dealing with data, which is the plural. We look at masses of information, and when we look at the data, we can come to conclusions. When you look at one point of information, one datum, all you know is that at that time, in those exact specific conditions, when this happened, that was observed. And it doesn't really mean very much. So you need data. You need large amounts of information. And when you can say that thousands of times this has been done in these conditions and this was the result, then you can say, well, gee, it's probably going to happen the next time we do this way. That's the significance of statistical analysis and large fields of data. And the reason you can reach those conclusions is when you look at large fields of data, you can say, well, it's very probable that this is what's going to happen. Or it's totally improbable that this is going to happen because you did that, because your data doesn't support that hypothesis. And so I looked at the idea of evolution from, as Dr. Harold Hill used to put it, from goo to you by way of the zoo. And I said to myself, this is totally improbable. It is statistically farcical to believe that this would happen. And if you listen, and I know I said I wasn't going to bash the evolutionists, but I'll stop now.
Father, bless these words to our hearts in Jesus' name. Psalm 139:14. The Word of God says that we are fearfully and wonderfully made, right? In the original language, what it's actually saying is that we are made in such a way that inspires awe. Inspires awe. We are awesomely made. Our design is such that it will inspire awe. And it really is. On every level, Doug showed you some of that with these molecular machines, so tiny you can't begin to see them, and yet incredibly complex. And we're going to talk about that complexity a little bit tonight for a few minutes. This is not something that we can cover in any great depth. You could spend years talking about this. But just to introduce the topic, it's called irreducible complexity. Turns out that everything in our system, every living system that there is, from the simplest to the most complicated, what we call complicated, what we call simple, is immensely complex. It has immensely complex structures, but it has immensely complex systems also. In other words, all these amazingly complex structures work together. If you take anything out, if you take a single thing out, the whole thing crashes. And so we're going to look a little bit at that. Irreducible complexity was brought forth by a professor named Michael Behe. He has a PhD from the University of Pennsylvania. He's a biochemistry professor at Lehigh University. On Lehigh's website, they have a disclaimer for him because he doesn't buy into evolution. Now, Dr. Behe was an evolutionist. He was a classical evolutionist. And then he started studying some of these molecular machines. And he found that there is profound design in these molecular machines. That if you take a single thing out, the whole thing stops functioning. Evolution is based on the notion that over time, all of these things improve. That they're functioning and then they function better. Something else happens and they function better still. Again, something else happens and they function better still. What Dr. Behe found was that you cannot have any function if you don't have the complexity that exists today. So people may say, what if you change functions? Well, now we're getting into hypothetical speculation. As scientists, we don't like to deal with hypothetical speculation. We like to deal with what is, not what might be or what could have been if. We like to deal with what is. Okay, now Dr. Behe made this statement. He basically, I'm not going to get into it, you can read it if you like, I'm going to summarize it. Basically says, and the first question I think is very good, what type of biological systems could not be formed by numerous successive slight modifications? That is the classic claim of evolution. Numerous successive slight modifications brought on by random chance mutations, each one producing a selective advantage. By selective advantage, I mean each one producing some characteristic in the organism such that it can survive in its environment and also its competition with other organisms a little bit better than it could before. Okay, what kind of an organism would that be? And then he said it would be an irreducibly complex organism. In other words, what doesn't or what isn't capable of experiencing that? Something that's irreducibly complex. Well, once you start looking at things and you start studying their makeup and their function, you find that virtually everything is irreducibly complex. Now, you may be able to simplify one slight part of something, but nothing works by itself. In other words, if I have a muscle twitch, that muscle twitch doesn't work by itself in order to become a muscle twitch. Something else is involved. As a matter of fact, many other things are involved. And so if I start picking things apart, I can say, yeah, this structure, maybe this structure, that nuclear pore, maybe if I took out what's called the nuclear basket, that underneath part, maybe if I took that out, you could still have a pore and maybe replace it with something else. You could still have a pore. Perhaps you'd have a pore, but would it be the pore that does what a nuclear pore does? Because nuclear pores have to be extremely precise. DNA is highly sensitive material. So if you start messing with those pores, you're also messing with the DNA. You're also messing with what the DNA does. And you start opening up a whole can of worms that is going to change pretty much everything. And so even though an individual structure, maybe you could alter it somehow, but put it together with everything that it needs to function, forget it. You can't alter it. Now, real quick, this isn't part of what I had planned, but it's coming to mind, and I think it's an important point. There's something called the rule of thirds. Perhaps Dr. Chung, you might be familiar with this, the rule of thirds. In other words, you can have 1 or 2 mutations that have no harm. The third mutation will have harm. Okay? That's how we started treating AIDS. There was a researcher at Johns Hopkins University a number of years ago, started applying the rule of thirds. Okay. AIDS used to be treated with 1 antiviral. That stopped working because the virus mutates very quickly. Notice it's still an AIDS virus. It's not now a giraffe. Okay. The virus mutates very quickly, and it's no longer sensitive to the drug. You introduce a new antiviral, the virus mutates again. You introduce another antiviral, the virus mutates again. You run out of drugs. This researcher came up with the notion of using the rule of thirds. In other words, give it 3 antivirals at the same time. Because maybe it'll mutate against 1, maybe it'll mutate against 2, but it cannot mutate against 3. 3 is the limit. Sort of a biological speed limit. You can get to 3 and you're done. Okay? 1 or 2, possibly. 3, forget it. Why is that? Well, part of it is irreducible complexity. Okay? Things are so complex that they can only work up to a certain point if you tinker with them. If you tinker with them by changing something, taking something out, or changing the structure of it, you're not just tinkering with one thing, you're tinkering with several things. Okay, you can tinker with 1, maybe 2, forget 3. Okay, irreducible complexity, definition. A single system composed of several well-matched interacting parts that contribute to the basic function wherein the removal of any one of the parts causes the system to effectively cease functioning. Classic example is right here. Okay? Your basic standard mousetrap. You remove any one of these parts, you no longer have a mousetrap. Okay? You remove the spring, 1 close. Remove the bar, 1 close. All of these parts are needed to work together in order for this mousetrap to be a mousetrap. Okay? Likewise, the flagellar motor, and that's what Dr. Behe was studying, was the flagellar motor, which Doug was talking about briefly. The flagellar motor is made up of all these really interesting proteins. Okay? These proteins all work together. They cross in different places into different levels of the membrane. The membrane is the covering of the cell. Doesn't matter what kind of a cell. They all cross into different portions of the membrane. Some can cross across the entire membrane. Okay, now, these proteins have to fit together just so. If they don't fit together just so, they won't work. Now, if you say change 1 portion of the protein, say 1 amino acid. Amino acids are tiny, tiny little things. Okay? They're the building blocks of proteins. Proteins are strings of amino acids that spontaneously fold in very precise patterns. You change 1 amino acid, you won't necessarily change the fact that you have a string of amino acids. You'll change the way that they fold, because the folds are based on positive and negative charges. If you change the way that they fold, you change the function of the protein. With protein, structure determines function. So if you change the structure even a little bit, you change the function of the protein. Usually you'll make it non-functional. Sometimes you'll make it dangerous. So if you change even one of these little proteins on this flagellar motor, let's say in this portion right here. Change one of those little guys and you're done. The motor will turn no more. So this irreducible complexity idea, this is not just some pie-in-the-sky idea that might work here or here. This pretty much works across the board. Okay, now, how complex is it to move your finger? Everybody scratch your face. Just with your finger, scratch your face. Was that really hard? Everybody got that? Did it take a lot of thought? Did it take a lot of effort? Hello. It's very easy, right? All right, so how complex is it to move your finger? Well, it turns out that it's irreducibly complex. Okay? The complexity involved is mind-numbing. And we're going to talk a little bit about what is involved in moving your finger. Okay? Now, I'm not expecting that you're going to be following what I have to say. Okay? Don't try to follow what I have to say. If this is the first time you're hearing it, it's going to be confusing to you. It was very confusing to me. Okay? But what I want you to get is an appreciation for the immense complexity that's involved in the simplest type of movement. Now, why am I picking moving your finger? For a very basic reason. Motion is one of the key survival elements to pretty much all organisms. If an organism can't move in some sort of a way, it's not going to survive. As a matter of fact, it's one of the 6 characteristics that determine whether something is alive or not, is motion. So motion is absolutely fundamental. And so we'll take a look at what it takes to get the simplest type of motion. Okay, these are the things involved. Again, I'm not going to get into all of them. You can read them for yourselves. But it starts with thought. Thought is so complicated, scientists cannot figure it out. Okay, at first they thought it's just a series of neural pathways and neural impulses and things like that. It's far more than that. The reason why they can't figure it out is because thought starts in the mind. The mind is not, you may not like this for a moment, some of you, but maybe you'll be okay with it. The mind is not a physical part of our body. It's not the same as the brain. Okay? The mind is one of the 5 parts of the soul. That's where thought exists. Now, the brain is basically the nexus, if you will, for the soul and the body. That's where the soul's initiations get converted into physical manifestations. So the mind is something we can't figure out. We try, psychologists try it all the time, but we can't figure it out. We can't figure out what really triggers a thought. And the reason is because it's not really part of our physical body. It's a part of our soul. So when the scientist can start examining the soul, he'll be able to figure out the mind. Okay, that's where preachers come in, right? Okay. Okay. Now, this is a structure of some brain cells, some brain neurons. And I want to bring your attention to just a few of them. I want to bring your attention to the cell body, which is this large portion here, to the axons, which are these long, stringy type things, okay, to the nodes of Ranvier, which are these spaces between the blue parts. The blue is myelination. And I also want to bring your attention to the dendrites. The dendrites are the parts of neurons that receive impulses from other neurons. Okay? The myelin sheath, we already mentioned, these blue things. The synaptic bouton, which is basically the end of the neuron. That's where the impulse is passed on to something else. And the oligodendrial cells, dendrial cells. Okay? Oligodendrites are simply cells found in the central nervous system, which is the spinal cord in the brain, that extend these projections, wrap themselves around the nerve cells. You've heard of myelination, right? You've heard of people like MS, they have problem with myelination. In the central nervous system, it's the oligodendria that do this. If the oligodendrite is not functioning, then the brain's going to have a lot of problems. Now, the oligodendrite does not actually carry the impulse. It's a support cell. And there are support cells in the central nervous system that do things like nourish the neurons or wrap themselves around to produce the myelin sheaths. If those support cells, which are not involved in the impulse, but if those support cells are not functioning and they themselves are complicated structures, if something is removed from, let's say, within the cell body of the support cells, maybe you remove something called the ribosomes, which perform the proteins. If you remove the ribosomes, these tiny little things, extremely simple, out of a support cell, the whole thing is shot. Okay? You're not going to get a nervous impulse even though it's not a neuron. You're going to get you could get a type of an impulse that'll be basically ineffective. So when we're talking about this stuff, we're talking about things that are very complicated, work with other very complicated things. Okay, real quick, the nucleus in the cell body. I don't want to go through everything in the cell body. I just want to go through some key parts in order for you to get an appreciation of what has to be in place just to move your finger. Okay? The nucleus. The nucleus itself is irreducibly complex. You start fiddling with the structure of the nucleus, you're going to destroy the DNA that's in it. It's going to allow all kinds of things in and all kinds of things out that don't belong there. Let me give you an example. One of the things that fiddles with the nucleus is OH groups, okay, known as hydroxyl in chemistry. Hydroxyl is what you find on alcohol. That's what makes alcohol, if you will, effective in the effects that it has. Okay, what it does, it puts holes in your membranes. All right? So that was akin to messing with the membrane, with the nuclear envelope. You start messing with that, you're going to start, number one, let me back up a little. If you start ingesting alcohol, okay, I hope nobody here does, but if you start ingesting alcohol, you're going to start taking on these OH groups. These OH groups will put holes in your cells. Holes in your cells, if they're brain cells, they don't replicate. Okay, that cell is done. Once there's a hole in it, it is done. Okay? Now, they don't stop at the cell membrane. They keep going right through the cell body into the nucleus. If they attach themselves to the DNA, and that's what they're targeting, if they attach themselves to the DNA in the wrong place, you get lovely things like cancer. Okay? The media loves to lie about alcohol. They say, well, it's good for the heart. I was talking to a cardiologist friend of mine a number of years ago. And I said to him, what do you think of all this stuff that the media says, good for the heart? He said, well, he says, flavonoids are good for the heart. You can get them in grapes. You can get them in tomato sauce. You can get them in pretty much anything that's red. Okay, that's good for the heart. He said, but it does triple your chance of cancer. Okay, the same quantity, that 2 glasses of wine they swear by. The media loves that. Why? Because they used to be a bunch of party animals in college and they want to justify their lifestyle. Okay? The media loves it. But the cardiologists tell us it's baloney. Okay. All right. Contains 4 specific nucleotides. That's another one. Nucleotides are those letters that make up the DNA chain, right? A, C, G, and T. Okay, change one of those letters, you're messing things up. Take one of those letters out, even worse, the whole thing will not fit together. Because the way that the chain aligns itself, it's dependent on the right alignment of all 4 letters. Take just one of the letters out, half of your DNA chain will not be able to align. Okay? Remember, it's 2 strands going side by side. They're connected with these hydrogen bonds. If you take just one letter out, say you take out T, ACG, you have a completely meaningless combination of molecules. Okay? Now, evolutionists tell us that this evolved, that somehow, they don't propose any explanations of how, they all say, somehow. Well, somehow is a very broad term. Okay? When you take one of those out, forget it, the whole thing is done. That's why you can't mess around with the nuclear envelope. In other words, the membrane that surrounds the nucleus. That's why you can't mess around with the pores. Because you start messing around with them, you start interjecting things into the DNA that just don't belong there. Okay. The membrane itself, extremely complex. Okay? The membrane is made up of a bunch of different molecules. We're not really going to get into it here, what those molecules are. Extremely complex. Essentially, the bottom line is it regulates what gets in and out of the cell. Okay? It separates the internal and external environments for the cell. If you mess with the membrane, you kill the cell. Or you introduce things in there that don't belong there, which can be very harmful. Okay. The mitochondria. Doug talked a little bit about mitochondria, the power center of the cell. Okay, I'm not going to say too much more than that. He said plenty, and it was good stuff. But I will say it depends on three extremely complex processes to function. Glycolysis, which is basically the breaking down of sugars into pyruvic acids. Then it depends on the Krebs cycle, also known as the citric acid cycle. Very complicated cascade of events. Okay, the idea is to build up protons. You remember he talked about a gradient. The idea there with the Krebs cycle is to build up protons on one side of that membrane that's inside the mitochondria. All right, and the third part is the electron transport chain, which is what that ATP synthase is part of. That basically, again, it's a cascade of events that takes place, pumping protons and electrons across the membrane to build that gradient of hydrogen that'll go down through that ATP synthase that'll churn to produce that ATP. It provides the energy for it. Okay? All of these things are irreducibly complex. The reason is because in each cascade of events, and these are three cascades, in each cascade of events, each event is entirely dependent on the proper function of the previous event. So the first thing must function properly in order for the second thing to take place. That must function properly in order for the third thing to take place, and so on. All of the structures within those are made of proteins. If you change a single amino acid on a single protein in that chain or in that cascade, you shut down all three cascades. So this stuff is highly complicated. If it's not functioning at one point, it won't function at any point. Okay, ribosomes, very simple little structures that are within the cell body. They're responsible for producing proteins. They read the nucleic material, the nucleic acid, in this case RNA. They read it, they interpret it, they synthesize the proteins according to the message. You tinker with the ribosome, it's only got 2 units. You tinker with the ribosome a little bit, the whole thing's done. You get no proteins at all. Okay? Proteins are the basic building blocks of every organism. You take out the proteins, you take away the organism. Okay. Now, the axon hillock is only found on nerve cells. The reason why this place is important is because it is the site of impulse synthesis or impulse initiation. Okay? That's where the nervous impulse takes place. It's called a depolarization wave or an action potential. Now, in order to move a muscle, you need to get an impulse that originates right here in this red region. It's called the motor cortex. Okay? That motor cortex contains these pyramidal type neurons. Right here in this little area at the base of this body, right? Oops, hold on, my hand's a little shaky. Right there is where the impulse is going to start. What takes place there is that little pores made of proteins, again, these little pores, they're pictured right over here. Okay, those little pores start moving toward that axon hillock. Now, they're not there all of the time, but they will start to move toward that axon hillock. When enough of them is present, these are sodium channels. In other words, they're pores for sodium to get into the cell or out of the cell, right? In this case, into the cell. They're sodium channels. Once enough of these sodium channels are present, then you have the ability to initiate an impulse. The initiation of the impulse will happen spontaneously just by the presence of these channels. Okay? Now, these channels, again, they're made of proteins. Change one amino acid, you wipe out the channel. Okay? So the notion that somehow these evolved, forget it. There's no way for these to evolve. They're either there or they're not there. If they're not there, you get no neural impulse. Okay? Now, these channels are also used in a lot of other things. Okay, so we're just talking about moving a finger. We're not talking about a whole host of other things. Okay. All right, we just mentioned a lot of what I was going to say about this. But basically, you have these 4 sections of these complicated proteins. The proteins, they're really hard to picture. When you see them, they kind of, in a schematic diagram, they kind of look like ribbons with strings attached. Okay, that's because there's so many different folding patterns to what is essentially a string that it manufactures these different patterns and shapes as it's folding, refolding. It folds over at least 4 times. Okay, you get these various different patterns of folding that make these different shapes. In Doug's presentation, you might have noticed one that looked like a spiraled ribbon. Okay, that's called an alpha helix. That kind of a pattern is not uncommon. There's also like a flat pattern. It's called a beta pleated sheet. That's not uncommon. It's all done by folding. The folding is all done by charges. The charges are determined by the order of the amino acids. If you change one, you change it all. Okay, it's all irreducibly complex. If you start tinkering with proteins, forget living systems. Okay, they just won't happen. The notion of evolving these proteins, well, again, you can't tinker with them. They're either there, present, completely functioning as they are, or they're not there at all. Okay, the myelin sheath. This is probably the most famous part of a neuron because the conduction is dependent on these myelin sheaths. If you have problems with the myelin sheaths, you're going to have problems with conduction of impulses, and you're going to get various unpleasant diseases. Okay. Now, myelin and saltatory conduction, real quick. Basically, it means this. Myelin is produced by one of 2 types of cells. If it's in the brain or the spinal cord, it's going to be an oligodendrocyte. Okay, what I showed you before. If it's not, it's going to be, like if it's in the periphery, like in your arms, the nerve cells in your arms, they're going to be produced by Schwann cells. Okay, one oligodendrocyte can myelinate 50 axons, 50 neurons. Okay, Schwann cells, one axon will have a bunch of different Schwann cells. Okay, again, these cells are fully complex cells. These are not simple cells. These are complex cells. Okay, that means they have all the complex stuff that all the other complex cells in your body have, plus a specialized function. Okay, now, when you have conduction taking place, scientists call it saltatory conduction. Basically, you have an impulse that's traveling across this neuron. This is the long part of the nerve cell, the stringy part. Okay? The impulse travels through that long part. Now, it's dependent on the exchange of certain types of atoms, ions, charged atoms, across the membrane where it's exposed. If you were to do that the whole way through, and some things do carry an impulse this way, there's a bunch of channels across an entire membrane. One channel opens, stuff comes in. That triggers the next channel to open, stuff comes in, triggers the next one, stuff comes in. If it worked that way in nerve cells, the impulse would be far too slow. And that's the problem with these myelination illnesses. The impulses are far too slow to be effective. So in this case, God designed it in such a way that the impulse only takes place in certain spots. And the rest of it, the charged atoms travel within the axon itself. That makes it a whole lot faster. Okay. Now, the axon terminals, those are the terminal boutons, some people call them. Nowadays, the more common name is axon terminals. They're found on the ends of the axons. The axons, again, are the long stringy things. Okay? Here you have several interesting structures. Okay? Basically, what happens at the axon terminal is you get the release of neurotransmitters that are sent to the next structure, either another nerve cell or maybe a muscle cell, but some other structure receives the neurotransmitters, and that structure does its own thing with them. Okay, now these structures themselves are fairly complicated, the process here. Okay? If anything goes wrong in here, the whole thing stops. Okay, this is how pain medications work, at least some of them. Some pain medications for pain impulses traveling back to the brain, they'll block the receptor sites. These are little receptors for the neurotransmitters. Well, as the pain impulse is being sent from one nerve to the next nerve, pain medicines, some of them, they come in here and they occupy the receptors so the neurotransmitter can't attach. If the neurotransmitter can't attach, the impulse will stop. Okay, and now you block the pain. That can work in both ways, both directions. It's a problem with some recreational drugs. You start messing with these neurotransmitters, you start blocking off certain types of receptors, and all of a sudden your nervous system isn't working. Okay. All right, the synaptic cleft. I just wanted to very briefly show you this. I'm not going to go again through all the different steps. You can read them if you want, but we're just going to talk about a little bit of this. In the synaptic cleft, you get these vesicles, basically these little sacs that are filled with neurotransmitter. They're sent down from the top of the cell where the cell body is. It's manufactured here, sent all the way down the axon, right back to here. Now, the synaptic vesicles, the vesicles in the synaptic cleft, they don't necessarily do anything until they're told to. And the way that they're told is there's a trigger that is calcium. It takes up calcium, that synaptic cleft comes in here. Okay, again, there's channels here, you can see them over here, whereby the calcium will enter there. Once the calcium is present, the vesicles will start moving to the bottom of the terminal bouton or the axon terminal. They'll fuse with the membrane down there because they're also made of membrane, and they'll empty out the neurotransmitter. Okay, now, that neurotransmitter will cross across this tiny little space called the synaptic cleft, and it'll get picked up by receptors. Now, the problem is, how do you then stop the impulse? Because if that neurotransmitter is at the receptor, you're going to have another impulse generated at the receiving end. In order to stop the impulse, your body releases another chemical that will break down the neurotransmitter. So you have all these negative feedback loops going on. If the negative feedback loops don't exist, they're not directly involved in creating the impulse, they're involved in controlling the impulse. So if you don't have these control mechanisms, you're not going to have a healthy organism. You're going to be able to clench your fist, but you'll never let it go. Okay. Once it gets through all its set of neurons, the impulse, and it finally hits your muscle. Okay? It's at a point called the neuromuscular junction. Okay. Now, the neuromuscular junction is not all that different from a synapse. It's a type of synapse, you could say. Except now, instead of two nerve cells, it's a nerve cell and a muscle cell. Okay? And again, you get these receptor areas right over here. This is called the motor end plate. You get the receptors on here. They start opening channels when the neurotransmitter is attached. In other words, you got a receptor, next to it is a channel. The receptor has a neurotransmitter attached to it, the channel opens. Okay, ions start flowing in, and just like with the nerve cell, once the ions, the charged atoms start flowing in, you get another impulse generated across the muscle cell. Okay, you go across the muscle cell, you get these nice little sodium channels over here. Impulse is going in, you got this nice flow of ions. Now the whole idea here is that these ions get down here into this portion. Okay, this is called a T-tubule, and there's a bunch of stuff that happens here at the T-tubule. Okay. Now, the T-tubule, also known as transverse tubules, are made of sarcoplasmic reticulum. That's not a real important name for this purpose. It's just what it's called. Okay, for our purposes, you just need to know it's a type of a membrane. Okay. Now, within this membrane, you've got all these calcium ions. The membrane basically crisscrosses across each muscle cell. Okay. Now, when I say muscle cell, when you see a picture of a muscle, you got a nice deltoid or a bicep or something, right? It's a good muscle. It's got thousands upon thousands of cells in there, countless cells. Each cell has an entire complete system of this stuff. Okay? So you get the release of calcium out of these basically membrane sacs that weave across the cells. That calcium has to be released into the cell in order for anything to take place. Okay. Now, once that reaches in here, okay, you got several things that become active. Okay, and the list of them is right here. We're going to go through them very briefly one by one, but this is the list right here. These are their pictures. Okay, the first one is the sarcomere. The sarcomere is basically a contractile unit. Remember, muscles contract. Therefore, you have to have little units within the muscles that are capable of contraction and expansion. Okay? Muscles are made up of countless of these little contractible units. They're called sarcomeres. Okay? The next thing you need is a protein. Again, it's a protein. It means you change one little thing in this protein, the whole thing is shot. Okay? You change one little thing in actin proteins in the way that your body produces actin, forget trying to move. Okay? You have this little actin molecule. It's also known as a thin filament. These green lines up here versus the brown ones, the thinner ones versus the thicker ones, those are the actin proteins. Okay? Here, they're pictured as these chains of balls. Okay? These are the actin proteins. Okay. Okay, the next thing you need is myosin. Myosin is the thick filament, also known as, that's this thicker part here versus this actin, this thick part. It's got these things on there called myosin heads. The myosin heads look like foldable golf clubs. If you could picture a golf club with a hinge in the middle of the club and then another hinge at the, whatever you call the part that you hit the ball with. Okay? If you consider two hinges like that, then you've got something that's very similar to this section of the myosin molecule. That's what does the pushing. That's the molecule that's responsible for the contraction itself. Okay, then you have tropomyosin. Tropomyosin is right here. Okay, and it basically what it does, it wraps itself around the actin. And the reason why it needs to do that is because you don't want these little myosin heads attaching to the actin, and that's how things happen. They attach to the actin and they pool. Okay, you don't want them just randomly attaching. So what that tropomyosin does, it wraps itself around the actin and it blocks off the attachment points. So it can't attach at the wrong time. Now again, it's a protein. Mess with the protein, you mess with the structure of the tropomyosin. Mess with the structure of the tropomyosin, you won't be covering the attachment points. Don't cover the attachment points, forget it. You're going to have a body-wide Charlie horse. Okay. Okay. Then you have troponin. Troponin is basically a little molecule that attaches to the tropomyosin, kind of acts as a handle. Okay? The tropomyosin, in order to be able to get a contraction, it has to move out of the way. And the troponin is kind of like a little handle that you pull on to move it out of the way. Okay? Now, what does the pulling on the handle? Okay? It's the calcium. Okay? You get calcium attaching to the troponin molecule, it'll shift the whole tropomyosin molecule, and now it exposes all these little binding sites. You see these little dark dots on the actin? Okay, those are the binding sites. Okay, when those binding sites are attached, now the myosin, the heads of the myosin, they're called cross bridges, now they can attach, and now you can get that contraction going on. Okay, now if you somehow inactivate the ability of the calcium to either be released, and everything that we talked up to this point causes that release of the calcium. The whole neural impulse, the purpose of that is to get that calcium released. Okay? If you take any one of those things that's involved in that neural impulse being sent all the way down through the nerve cells, through the muscle, into the transverse tubules to release the calcium. Any one of those things gets changed or removed, the calcium will not be released, therefore you will not get a muscle contraction. Okay? Now, once that happens, these sites get exposed. Okay. Now, the exposing, this is kind of what it works like. I don't know how long these will continue to run. For some reason, they only run for a short time. This is what the contractile unit does when it contracts. Okay? You get this kind of pooling, and as it's pooling, it slides one filament, one string, one molecule past the other. Okay? In causing it to slide past the other, you're causing these things to contract, these units to contract. Okay. Now, it's a fairly straight up process. I'm not going to get too far into it because, again, you can read it for yourself if you want. But basically, you take one of those ATPs that Doug was talking about, and that causes the pulling back of that cross bridge, of this little myosin head. It cocks it back like on a gun. Okay? Now, when those binding sites on the actin are exposed, this is attracted, comes back up here. Okay? And then, because it does have some energy, it'll cause a pulling motion. Kind of if you take your arm and bend it with your wrist. Okay? It'll cause a pulling motion. Okay? But it's pulling across something. All right? Then another ATP molecule comes in. Okay? It gives it the energy to disattach, pulls it back in a cocked position. This can happen thousands of times in a second. Okay? Now, this whole thing, from A to Z, is repeated hundreds of thousands of times in doing this. Okay? It's immensely complex. Okay. Structures involved, we're not going to get too far into this. But real quick, skeletal system, bone cells are amazingly complex what they can do. We don't have time to go into it, but those themselves, without bone cells, without skeletal system and all that does, forget trying to contract anything. You're not going to be able to move anything. The whole purpose of the muscle is to move the bones. Okay. Okay, circulatory system, you don't nourish those cells. If you don't have that circulatory system present, the cell's going to die. If you don't give it oxygen, it's going to die. Oxygen has one purpose, to produce ATP. It's the end part of that whole cascade to produce ATP. Okay? Otherwise, oxygen is quite poisonous. Okay? So it has one purpose in the body, and that's to produce ATP. It goes through the blood. We have 2 seconds of stored ATP in our bodies. Okay? In other words, we are working on a 2-second energy reserve. Okay?
That explains a lot.
Yeah. Coffee is trying to fix that, yeah. Okay. Respiratory system, again, another immensely complex structure, immensely complex processes, obviously for getting oxygen to those cells. And then the brain, if you don't have the thought, if you don't have a properly functioning brain cell to interpret the intentions of the mind, to cause the right type of impulses, you get nothing. Okay? So we indeed are fearfully and wonderfully made. Our whole design is awe-inspiring. Okay, that's it.
So technically, we're 4 minutes short of when we were going to stop the presentations and just spend the rest of the time in Q&A. But we still have one more presentation, and it's one that I very much want to hear myself, and I'm sure you do too. So let's take about 5 to 10 minutes tops. That'll give Dr. Chung a chance to get wired for sound and to get his presentation loaded into the little well-designed machine that we're using here.